every living thing on Earth has a genome made out of the same 4 DNA bases: A, T, C, and G. It's the different number and order of those bases in each organism that gives us all of the variety of life that we see. And even within a population of organisms from a single species, no two organisms are exactly the same. Individuals have different genetic traits as a result of small differences in their DNA sequences. These differences make up a population's genetic variation. In this video, we're going to talk about what genetic variation is, how it arises, and why it's important. Genetic variation is a term that covers differences in gene sequences between organisms. The two main sources of genetic variation are sexual reproduction and genetic mutations. So let's talk about mutations first. Mutations are really just typos in the DNA sequence. So here we're looking at a strand of DNA. A mutation could be a swapped nucleotide, like an A instead of a C, or a small insertion or deletion of base pairs. This can happen when cells replicate their genome to make new cells. So they make copies of their DNA by carefully pairing each base on one side of a strand of DNA with its complementary base. And this is usually super accurate. But about once in every 10 billion base pairs, a mistake can happen. And those mistakes can change the code or expression of genes. And that can change a phenotype. When these mutations occur inside the coding region of a gene, they can create a new version or allele of that gene. Sometimes the environment can cause mutations too. So things like UV radiation or X-rays and certain different chemicals can damage DNA. And when a cell repairs that DNA damage, it can sometimes make a mistake and add one of these small mutations. Mutations that happen in one cell can be passed down to any new cells that result from cell division. But to be passed on to a new generation, like from a parent to a child, a mutation has to happen in a germline cell like a sperm or egg cell. These mutations, if they result in a phenotype and are passed on to a new generation, can add heritable genetic variation to a population. The other main source of genetic variation is sexual reproduction. By combining DNA from two individuals, alleles get shuffled together, creating new genetic combinations. This rearrangement comes from two main processes: meiosis, which happens in each parent individually, and fertilization, which combines the DNA from two parents. Meiosis is important because it creates genetically unique gametes, like sperm or egg cells, from the genome of an organism. Many organisms, including humans and other mammals, are diploid, meaning that they have two copies of every chromosome, one from each parent. These are called homologous chromosomes. And while those chromosomes each have the same set of DNA and genes, they can have different alleles of those genes. But for reproduction, each individual gamete, so each sperm cell or egg cell, must have only one copy of each chromosome, making it haploid, so that when two gametes come together, the resulting organism is diploid again. This process of going from diploid to haploid results from meiosis. Meiosis involves 2 rounds of cell division. In the first, called meiosis 1, each individual chromosome is replicated so that the cell now contains 4 copies of each chromosome, 2 exact replicas of each parental chromosome. The 2 replicas, referred to as sister chromatids, are connected at their center. So if we look at these chromosomes here, you can see that this chromosome was now duplicated. So now there's this additional chromosome, and these two together are called sister chromatids. And the same thing has happened for every chromosome in the cell. In the next step of meiosis 1, the homologous chromosomes find one another in the cell and line up so that each copy of a chromosome is next to its other copies. So the pairs of homologous chromosomes, like here, get really close together. And these adjacent chromosomes can actually swap small pieces of their DNA in a process called crossing over. So these regions that are super close together can actually swap. This means that a version of a gene that originally came from one parent, like this blue chromosome, could now reside on a version of a chromosome primarily from the other parent, like this yellow chromosome. So that'll look like this with these regions of swapped DNA. This will eventually increase the diversity of genetic variation found in the resulting gametes. So next, the chromosomes align in the center of the nucleus with homologous chromosomes side by side, and one set of sister chromatids from each pair is pulled to either side of the cell. So these ones are pulled to this side, and these ones are pulled to this side. Then, when the cell is cleaved in two by cytokinesis, each resulting daughter cell contains a haploid genome, but with duplicated copies of each chromosome. In meiosis II, these daughter cells are split one more time without DNA replication. So the sister chromatid pairs line up in the center of the cell, and one is pulled to each side so that when the cells split in two again, the resulting cells each contain a single haploid genome with one copy of every chromosome. And as you can see from our example here, each resulting gamete cell has a different mixture of alleles, both from each other and from the original parental chromosomes. This comes from both the crossing over that we mentioned before and the fact that the sister chromatids were randomly sorted into the daughter cells. So this increases diversity in how DNA is passed from generation to generation. Genetic variation is further increased when two unique gametes from two different individuals come together in fertilization. This keeps each new individual genetically unique. And this incredible genetic shuffling, reassortment, and recombination results in genetic diversity that can help populations adapt and respond to environmental pressures.